um, will be coming into more details about what's actually the platform is and how to work with it. So this is what it looks like, the front end. Now it's been up for quite some time. You can locate the front end to the data platform at data.SciLifeLab.se, and it's a common front end to have a good findability of different services. And we would like to interact with the entire community around the data platform, and this sort of launch event is one way to create a little bit of go out and introduce this and make sure that we get a good start toward the community with this. So as a background, for those of you who come from elsewhere, I mean, the SciLifeLab is a national infrastructure for life science and has been around for quite some time. And there are several parts of what SciLifeLab is doing. The infrastructure part, SciLifeLab provides infrastructure services in life science since many years back, and has 10 technology platforms and 40 different units that provide different types of technology services to the life science community in Sweden. But SciLifeLab also, since a couple of years back, is organizing the Wallenberg Foundation funded program in data-driven life science, which is there to essentially transform Swedish life science towards a more data-driven way of working, and new types of research, and also boosting the capacity in the country to work with data in life science and increase the capability for technical components to the life science area. And our role in that, in the data center, the philosophy behind what we do is, data center is a part of the central SciLifeLab organization and not one of the platforms. We try to do make sure that the programs, the different parts of SciLifeLab, has good support for a research data environment based on the FAIR principles, open science, community engagement, and this is linked very tightly to strong IT infrastructure thinking and a production think that the services should be well maintained. We think about the information security properly and handle all these things so we have a good environment with well organized and everything that needed to do stuff long term for technical things, life science. So our aim is really at the data center. We work together with several other universities around the country in the DDLS program for different scientific areas. The DDLS program covers cell and molecular biology, precision medicine and diagnostics, evolution and biodiversity, and epidemiology and biology of infection. Each of these areas also operate a data science node that works together with the data science in developing data services for the country. So the idea behind this is that we should work towards a maximum impact of the data and the technical work in life sciences in the countries to have the good impact for this. We should work to create this national work. It's not something that we drive just centrally. We build this and do the good for Sweden, work entirely nationally. We think that also increasingly we can see that the services, the development of technical work from the community is going to be incredibly important. So the role for us is to support the community to actually be able to develop and contribute and make this available at the national level in the best possible way. And that's why we created the data platform, to provide that type of environment for the community. We do this together with actually infrastructure collaborations with, for example, Linköping University, with the Berzelius system, with different other universities providing infrastructure, and NICE, which is the national infrastructure for research in general. We have our own international collaborations, for example, with VIB or EMBL. We work with integrating AI methodology in our services. We work very much about training and community building for technical staff. We try to take care of technical staff in our own organization, but also in the entire community, to make sure that the tools are available to work in a really professional way with data and IT for life science. It also provides the link to the infrastructure and data producing facilities in SciLifeLab for this. So the data platform in general, I mean, it's a fussy word, like I said, but what is it? It's essentially something that enables the technical environment with support services, making it possible for users wherever you are, could be researchers, mathematicians, engineers, but also from collaborators in healthcare or industry partners, to work with data that comes either from own research projects or from data sharing platforms here, or imported from other parts of the world, or produced by SciLifeLab platform, for example. And this technical environment is hosted and built on top of the infrastructures that we build at the data center in collaboration with the main infrastructure providers in the country. So this consists of a number of components. So we are supposed to work with all these different things that help provide functionality for the community. And this is really for the community. You can see that it's a production service environment for the community, with sort of for developed life science services. So now, very often, you know, if you have in your research groups or at some of the units, infrastructure units, you may have data services or databases or web services or tools that are built, and once something is built and provided, the running of it and the long-term maintainability of it could be a little bit difficult, because sometimes you have to work from a server on your desk or in the wardrobe, or setting up infrastructure hosted elsewhere. So one part of the SciLifeLab Data Platform is to provide a good hosting environment on the national level, where as a provider of services, as a developer, you would have access to compute and storage to run your things inside this platform, with a full credit to each provider, each service fully supported by the different providers, side by side with what's coming from the community inside SciLifeLab and outside. The infrastructure is supported by SciLifeLab and the DDLS program, and we're aiming for full transparency and reproducibility through Open Science Fair. We want all the stuff coming out of this to be as open as possible, and also working with that the compute parts, the code should also be open as far as possible. Some of the services coming from the data center that's been developed at the data center is also hosted and findable through the data platform now. For example, the pathogens portal. This has evolved from the National Swedish COVID-19 data portal to provide the sort of aggregate data support tools, services for the infectious disease community. And this is now found at pathogens.se and linked to the European pathogens data platform with the EBI, very close collaboration with EBI. We have the data repository, which is a kind of general data set repository, free for all Swedish life scientists. It's not a storage platform, but it's a publishing platform. You can publish data sets composed of essentially all data types. You will get a DOI. This will work very nicely if you publish data, for example, to journals, supplemental data, any kind of data set that you would want to publish in a more permanent way. You can also keep data sets under embargo or open for reviewers only. And you can publish access instructions and metadata if you have sensitive data. The SERV is another system provided through this, linked to the data platform, and it serves PyTorch or TensorFlow models, for example. You can host Dash or Shiny apps on this and other compute applications. So this is one way to kind of keep that linked. It's accessed also to the CPU, GPU, and storage resources we have. We have the research data management support done together with NBIS, and essentially that's what I wanted to do just as an introduction, like this little overview. We have now examples, and Lian will go into the more details about what the platform is in more detail. So that's from me. Thanks.
Thank you very much, Johan. That was great. Very good introduction. So as Johan said, I will kind of be taking us through a little bit about what the platform is, and how it looks, and what it can be, you know, in terms of use to you. So I will start sharing my slides. So hopefully you see my slides now. Someone screech if not. So yeah, I'm a project leader at the SciLifeLab Data Centre, and one of the projects that I lead on is the SciLifeLab Data Platform. So what I'm going to show you today is a little bit more about the services we have and the hosting resources, just to elaborate a little bit more on what Johanna said, and to kind of guide you when you're kind of trying to navigate this data platform. So a little bit to kind of just fill in on the background a little more. So we had our soft launch in the summer of 2022. That was when the site itself first became live. And over the last year, or maybe just less than a year, we've been working with people in the data-driven life science community in Sweden. And that means all of the affiliates, MDIS, all of those different sorts of organizations, and individual researchers too, to see what content exactly we should have on this, what would be useful to the community, what does the community feel like they need out of a resource that they don't currently have. So we've been having the researchers as our main target audience. So anyone working in data-driven life science in Sweden is considered, you know, the researchers considered a part of our main target audience. This doesn't mean that we don't accommodate for other types of user. For example, we often found that with portals and platforms that we've developed before, we get a lot of policymakers coming in to see kind of the latest situation, and also journalists aiming to promote research often use it too. And so this isn't like a closed space. So whilst we do have the data-driven life sciences program at SciLifeLab, and they're an incredibly important part of this community, the platform is not just for them. It's for a much wider audience than that. So, you know, please, this is a very inclusive space. Don't feel like that you're excluded in any way just because you're not specifically part of that program. What we ultimately aim for is that we have this the data platform as like a one-stop shop almost for data-driven life science resources in Sweden. That means if you're looking for a resource, hopefully you should find it here. And you should have everything linked to this. So this could be, for example, advice on where to find compute resources that you need. It could be the direct services that we link to. It could just be the latest in what's been published in data. So hopefully you should get this. So our specific aims for this will be to provide access to and hosting for appropriate services. And by appropriate services, we'll talk more about the requirements later in my talk. But basically what we're aiming for here is to have data-driven life science resources in Sweden and be able to access them and host them from here. Obviously, some resources will have limitations, and we will describe that where that is in terms of access. And one new key thing is to promote data-driven life sciences research as much as possible. So there's various ways that we do this, and we want to get everyone more aware. Obviously, publication of research is just one way to be able to do this, and we hope to be able to do that a lot more in future. And resources related to community building and support, we've built those into the platform because we've seen, again, from previous work, that what we've had when we've got this community built around a resource is not only does the research kind of resource improve over time, but we also have a constant flow of information between that community, so between members of that community. And that means that research can progress a little bit faster, everything can run a bit smoother, and that's ultimately great for any kind of research area. We also want to provide guidance on best practice for researchers. So we link to a lot of resources, for example, that detail that best practice, and we also provide direct advice and guidance where possible. So as Johan mentioned, this is very much a platform of two halves. We have a website, data.SciLifeLab.Se, and this is basically what you'll see when you kind of come to the page and you land on it. You'll see exactly this, and this will be the interface that most people kind of work with and are familiar with, and I'll go through this and like what's available on it just in a second. And we also have an underlying technical environment for hosting. So it's very much two separate things that kind of come together as the data platform. So just to kind of lead you into the website, obviously, like I say, you can go into that homepage, and we have the services. So you can access the services either directly from the homepage where it says SEAL services. We only show a very small subset of the services we have available on the front page. And if you dive into our services section, you'll see that there is a division between services intended for research, and services intended for data producing units or facilities. So both are accommodated for in this space. Excuse me. You can search for the keywords for those different types of units. So each one has a search and a filter. So if you're looking, for example, for a compute resource, you'll see that there are links to the compute resources that are available as services here, or other information. So you can kind of filter everything and go through. Hopefully this will guide you more clearly to the resources that you want to find. And it's possible to add yours. So if you have a service that is not featured yet and you think it should be, because it provides value to the data-driven life science community in Sweden, then please get in touch. We have recently added a lot of services related to MBIS. So this section is expanding all of the time. On our homepage, we don't actually show very much information beyond a small description, because we're hoping that people will lead in and explore these services in a lot more detail. So the full list of everything. And as you can see here, what we have is a small description, a link title, what type of resource it is, who it's maintained by, and quite importantly, support. So if you're looking at that resource and you are interested in getting engaged, or you need support in any other way, then you can kind of go here, click through. You can see their GitHubs as well where appropriate, and their web pages. So it's a very direct way to get support for your research. So that should give you kind of a key into how you can use this before you kind of click through to the services themselves. So just some example services. So Johan already mentioned the pathogens portal, or what was formerly the COVID-19 and pandemic preparedness portal, is just transitioning over. So I'm just going to kind of use this as an example of the types of things you might find in services. So this, for example, is a service that provides best practice. So it provides a lot of information on fair and open science. And as Johan mentioned, this is really key to the ethos of Data Centre. And it's also key to every service that we have listed on the services page. So they have a commitment to being as open and fair as possible. So they are all adhering to that commitment. Or supporting researchers in making their data more open and fair. We have a lot of dashboards on there that visualize data. So this is a way to click through into data resources that you might get access to if you're interested in infectious diseases data. There's a lot of information there, and it makes it clear with the visualizations that are custom. So you're not finding these anywhere else. And it means that you can kind of go through, get code, for example, off of these resources. We also have a large commitment to promotion here. So those sorts of things are really important, and links to other resources. So for example, one of the key resources on the data portal here, the pathogens portal, is that we have the biobanks resources. So basically a massive list of all of the COVID-19 resources that are available through biobanks is available directly on the site. But that's not the only kind of resource we can have. So we're also bringing forward the SciLifeLab Data Repository, for example. So this has a lead into helping researchers make their data more open and fair. It is powered by Figshare. It's an institutional instance. The data and items that are submitted are curated by members of SciLifeLab Data Centre and MBIS. So they come in and we make sure that they're as open and fair as possible before publication. So it's really kind of showing this commitment very keenly. You can publish your data openly, but if there are restrictions in being able to do that, then there are, for example, embargoes available. So you can delay publication until that your paper is published. And you can also have, for example, help from the data center to manage those access requests. So tell people what you need for them to submit in order to be able to kind of access the data, and then have that managed through us or through your university, depending on your precise situation. It's not intended for data that could go into a subject-specific repository. So in the aims of fairness and being open, anything that's intended for a really subject-specific repository would be intended to be found in that subject-specific repository. But it does allow a lot of item types that don't go anywhere else and a lot of data types that don't go anywhere else. So it's incredibly useful. For example, at the end of the launch event, I'll be making the slides available through this repository, so that everyone can access them, as well as hopefully the recording. So this is a completely different type of service. As I mentioned, please look through the services that we have. We have a considerable number of, for example, information about ADA Data Hub is listed there in the service, Metabolic Atlas, Human Protein Atlas, for example. There's loads of different things that are available there. So it's really key that you kind of look in and dig around and see what's there for you. We've tried to accommodate as many people as possible. So indeed, promotion for the services does not end at the service page or even during this talk. We do promote it on social media. We provide information about recent updates where that's possible. So, you know, those services, if they have recent updates, are a way that we can do a more speedy update. So, for example, we mentioned the data repository, that we put items that are published in there right on the front page. So you can click right straight through the latest information. And we have, for example, the service that Johan mentioned, SciLifeLab Serve, that hosts Shiny apps and Dash apps, as well as machine learning models. They can all be accessed directly here from the front page. So we will put a lot of effort into, if you have a service with us, into promoting it and getting people in so that your service gets the attention it deserves, because we believe that's really important and we want to raise awareness of what's available for the community. Speaking of community, we have community building as a strong commitment that we have here. So we have these small articles called data highlights. So they are used to show research that is openly sharing resources. So this can be code, this can be data, for example. They're either written by the researchers themselves who are involved in the work, or they're written by members of the platform team, and then they are go through an editing process. And we have a few talks later about users that have had their work turned into a data highlight before. So I don't want to labor on that point. But this is a really good way for getting your research more attention. They are shared widely in news. They get a lot of attention when they are released, and we will promote them wherever possible. So it's a good way to kind of get a lay summary of your work. We also have advertisements on the website for jobs, for example. So if you have jobs that are available in the area of data-driven life science, please tell us. We have forms to suggest about, you can make suggestions about open positions. We're always keen to put those up. We also have sections on events and training, so we try and keep up with everyone's events. So please inform us of those. And obviously, then people coming to the platform will see those events and see the training, and then they will come in. And we also have calls and potential funders. So one of the user cases, for example, that was brought to us is that researchers new to Sweden don't necessarily know all of the funders and how they can access funding for this area. So it was important to show not only calls that are like now available and open, but also potential funders that they should look out for. So that has been added to the page too. We update these at least every 2 weeks. Quite often it's more like weekly. So if you have a job or anything that's going out, we will also post all of the jobs that we're aware of on here. So hopefully that should give you a little bit more promotion, a little bit more attention for those things. So please get in touch. So second half of my talk is all about the hosting. So we have hosting, or provide hosting on the data platform with data-centric tools, databases, and other types of resources that provide value to the wider community in Sweden. So in no way is the hosting meant to be a storage system that the other one singular group can use. There are lots of other resources for that, compute resources that are very valuable to be able to do that. This is a place that can expose to a much wider community. So it has to have a wider benefit than one group. We don't expect any kind of particular credit to be assigned for providing the provision of the hosting services. And we provide them free of charge within our available resources. So we have funding from SciLifeLab and POV for these things, and from SciLifeLab. So those things within limits are provided free. And they have a few different requirements. So for example, like Johan mentioned in his talk, we require that all of the code and the outputs are published under an open source license. So this is to encourage our ethos of fair and open data sharing. So we really have that at the heart of this too. The resource must be containerized, which just really means it can be deployed on the web. We will need information about approximate usage statistics. Obviously, if your research has previously been, if your service, has been previously hosted elsewhere, you'll have this. If you don't have this, then an approximation based on intended use and things like that will be really useful, and information about how it can be set up. Other requirements include that the service must be actively maintained. So if it's just a resource that was put on there and is no longer maintained and no longer intends to be maintained, then obviously that's not the most value out of that necessarily. So we require that everything is actively maintained for the wider community's benefit too. And they should also have regular security scans. So we require that there is this regular thing to make everything remain secure. So that basically if it loses its kind of secureness, then we can kind of advise on how that could be amended potentially in some cases, but not always. It will be up to the user and the provider of that service and what's hosted to maintain that security. And that means that if it loses that, then we will unfortunately have to remove it if it cannot be resolved. But we do ask for contact persons so that we can get in touch in those sorts of situations. There is lots more information about hosting and how you can apply. So please click the web link here when the slides are made available to you, and then you should be able to access everything and read it in more depth. So you don't have to remember everything I just told you. So we're already making plans for the future. Like I say, we've been developing this for the last year, but in no way do we see this as a static resource. So it'll be constantly developed and improved, and we welcome suggestions and comments at all times. So one of the things that we have planned for the future is a bit more of a resources section. So we basically have, obviously we have our services, and people can click through and get information about that. And those are very valuable resources for the researchers. But we realized that there was this need, it came in from a few different users, of having information that's just not necessarily a service in itself, but is very valuable to the researchers for other reasons. So for example, we have right now listed as a service a list of compute and storage resources in Sweden. So we have information about what is available, how you can apply for access, how you can get through to that. And we'll continually expand on that and improve on that over time. So anyone looking for compute and storage resources, for example, can go straight there and see what's available within Sweden, what might be appropriate for them to use. We also are drafting information on how to maximize your alternative metrics. So for example, we've noticed that there will be this drive, an increasing drive to having researchers show their alternative metrics or alt metrics, not just their standard publication metrics, to be able to achieve getting funding and promotions and all sorts of things like that. So we're kind of making resources available to be able to kind of guide the community on how they can already start working increasing their alternative metrics. And obviously this section, like all sections, can be expanded upon request from the community. So if there's something that you wish we had that we don't currently have, please always feel free to get in touch and let us know. So a little bit of a recap, just to kind of finish us off and to show that we are meeting all of the aims I told you about in the beginning. So we're hoping to act as this one-stop shop for data-driven life sciences. So we have access to and hosting for appropriate resources. This can be found in our services and hosting section, for example. We have promotion for data-driven life science researchers, again, in the services, because we have a lot of research involved in those services, and in our data highlights. We also have resources, a lot of resources related to community visiting. Again, the data highlights, jobs, the funding, and the events and training. And we are providing guidance for the researchers on best practice. So we have a services section again, events and training, and our future resources section that we can expand on and provide. So just to kind of quickly give my acknowledgement. So funders, community, MBIS, who've been great during this, and team members, both past and present. I couldn't possibly list everyone here. So if you want to learn more and contact us, please find us on data.SciLifeLab.de. You can get in touch with us via our Twitter and LinkedIn, and we have a contact form on the website should you wish to learn more. So that is the end from me. I think we have time for perhaps a couple of questions, if there are any in the chat. Sorry, I couldn't get a chance. Okay, so perhaps we will move directly on to the first of our user talks and save questions for the end. So my pleasure to introduce Wesley Shaw, who's a researcher at Upsala University. So please, Wesley, take it away.
Okay, thanks. With this. And here. Okay, I've been asked to talk. You see, I've got mute, not mute or anything. Good. I've been asked to talk a little bit about a project that we had going, and that has been identified as in the data highlights. So this is for migrating long read sequencing to clinical routine screening. So I should say this is a collaboration between parts of Uppsala University, myself, Adam Amir, and Ola Spjut, advisor, probably one for our department, and the hospital, which I've seen most recently called Uppsala University Hospital. I know it as Akademiska. So the basic background is CML, chronic myeloid leukemia, sometimes different names, of a crossover to fusion protein. I'm not going to go into the biology very much, but just talking about one, the Philadelphia chromosome. The most important part is that for us, for data science, is that we'd actually, this gene isn't very big, so we can sequence the entire thing all in one shot and see what the mutations are in this particular fusion protein. Try to help with diagnosis of various types of CML. So what the hospital was doing before was the classical Sanger method of finding out different blips over there and assigning them to them. And the process takes a little while. There are some ambiguities in the whole thing, and try to move that. We want to try to move that into the NGS method, but actually more specifically long read method for doing the sequence analysis. And as I said, because since the process is actually short enough to be able to fit into one long read system, we can find out everything about that. And we find out information that is very hard to find, to determine through either short read or Sanger classical methods. So the first thing we did was, well, a couple of other preliminary studies. First thing was big published was a comparison study. So figure out for a small number of samples of patients, be able to test by classical method, the Sanger method, the number of samples that found in particular these mutations they're looking for, and then found by our method, we found everything that Sanger could find, and we found more things that it couldn't find, either because they're too low percentage of that particular mutation, or just whatever, it's not there. We never found any, we didn't fail to find anything that Sanger could find. So we could reproduce the results and potentially give more information. So what do we have? Here's the main, very, very simplified workflow. Is at the hospital, they take samples, send it to universities for sequencing. And then in this little dotted line here, this is the system that we created. It was to automate the processing because there weren't things that are very commonly used tools for the long read sequencing. They were for NGS, but not for the long read stuff. So we made these tools, made them all quite automated, push button, and send off the results to a database, a server that we're controlling, and have a GUI, a front end for the clinical people to use, clinical researchers, doctors also. What I mean by clinics, actual doctors. And the important part of our system, I think it worked quite well, is that we made it for them and in collaboration with them. I want to say, what kind of things do you need to see on the page? What way do you need to look for the information? And that, I think, worked out quite well. We have our workflow, won't go into detail of that, but one thing we can find that's hard to find through other ways is times when there's mutations on the same sample. You could tell if it's in the same sample or if in different samples. This you could tell if there's multiple mutations. So results, very simple interface. I've actually removed a number of columns out of here, partly for privacy, because these are humans, and just to clean things up a little bit. Is they use a search form, regular search form. They look through by dates or by mutation or by particular code names or whatever thing they need to look for, and find out what mutation do they see. With more intense color means the higher rate of mutation, of course, just color coding by that, heat mapping in a certain simple way. And they can get down to more details of finding this is the sequence we were looking for this mutation. Here's the number, do we find it, or is it negative? All the different things we're looking for, and many other types of information displayed for them. Won't get into detail. So altogether, we've got like 800 clinical samples. So they accepted this into their routine methods, and it was their normal way of doing things. So considered to be very successful campaign. That was just a real quick overview. If I said it quickly enough, so I wanted to show.
Yeah, great. Thank you very much, Leslie. That's really great. So I will move directly on to, because that's perfectly timed, to our next speaker, who is Ailey from Ender. And you take it away.
Thanks. I'm here to talk about SciLifeLab Research Data Management Guidelines, or RDM guidelines at short. So this is a knowledge hub for the management of life science data in Sweden. And it's found at dataguidelines.scilifelab.se and is built and maintained in close collaboration between SciLifeLab Data Centre and NBIS. So it contains written guidelines and best practices for the organization, storage and preservation, and sharing of data that is collected or analyzed during a research project. And one can find relevant information by exploring a phase in the research data lifecycle. So by clicking on one of the phases in this data lifecycle wheel, you will get an introduction to that phase of the lifecycle, what data management tasks to consider, including information on relevant resources and training material. You might also need guidance on a specific topic. For example, during this event, we've heard a lot about things needing to be fair. So if you don't immediately remember what the FAIR principles stand for, you can visit the RDM guidelines and click on this topic to learn more. Or if you are starting a new research project involving human data, for example, you might want to read more about that topic. The RDM guidelines also serves as the help desk for SciLifeLab data management support and as an information or advertisement space. And this can be found mainly in the boxes to the right. So if you are in need of more hands-on data management support, like writing a data management plan or finding a suitable repository for data sharing, you can contact us via this form and request support, or send an email directly to data-management@SciLifeLab.Se. So it's really great that the RDM guidelines can be listed as a service on the data platform. It increases our visibility in general, but especially helps us to reach our target audience. And also the team behind the data platform is very responsive and easy to work with, which I really appreciate. So before handing over to the next speaker, I would also like to mention that the RDM guidelines is a work in progress, and that we would like this to be a community effort. So for the community, by the community, contributions are much appreciated, both in terms of actual content, but also suggestions for improvements. So I would like to take this opportunity to thank those who contributed so far, and look forward to seeing this contributors page grow with time. Thank you.
Great. Thank you, Eilin. Okay, so we'll again reserve questions for the end, but I would like to introduce Oliver Bilker, who we're very grateful to have joined us from Arlanda Airport, I believe. Oliver is a professor at University. So please, Oliver, take it away.
Yes, thank you very much. I got started here last night and I'm still here. So my connectivity has deteriorated a little bit for the last few minutes. And I hope you can all hear me. I will perhaps turn my camera off.
We can hear you. You're fine.
Does it work? Okay, but I stopped my video just in case. So here we go. I was very grateful that the data portal highlighted some of our recent research as a data highlight. And what I would like to do now is just give you a bit of context, how that fits into our broader research and how I would really like to use the data platform in future. The project that we work on is on finding functions for all malaria parasite genes. That's the Plasmodium Genetic Modification Project. Our target audience is really, in the first instance, the malaria research community worldwide. We're migrating this project from the Wellcome Sanger Institute, from where I moved 4 years ago, to Umeå, where we have the molecular infection medicine Sweden, an institute that's the Nordic Swedish EMBL node. So the work we do, the work we did in this particular paper that came out in February and that was featured, there we asked how sex is determined in a malaria parasite, and we created various types of data in our lab and with the help of SciLifeLab platforms. One data type came from a systematic screen in a rodent malaria parasite model, where we asked which mutants that were equipped with barcodes were required for making male and female sexual stages, and these are important to transmit the parasite to mosquitoes. And the green and red genes there on the left were the hits of our screen. And then another data type involved analyzing these hits better. For that, we needed to create a detailed transcriptomic atlas of sexual development, and that's shown here, this developmental trajectory from the asexual cycle that makes people ill to the male and female transmission stages that infect mosquitoes. So we used 10x data to create that. And then we knocked out the top hit genes in our lines in our parasite and made clonal mutants and used SmartSeq2 transcriptomics to characterize a few hundred cells of each mutant. And we mapped these cells onto the relevant part of the developmental trajectory in our atlas. And with that, we learned a lot about, we used the transcriptomes to stage these individual parasites, but also to learn about how their transcriptome is specifically perturbed. And through analyzing all that data, we came up with this hierarchy of genes that affect sexual development, and we published that, and many colleagues in the community are now picking up these hits to do in-depth molecular characterization of the mechanisms that are important for initiating the transmission stages of the malaria parasite. Now, this is part of a much bigger project that aims to assign functions to all malaria parasite genes. The reason we do that is because much malaria research, important malaria research, is genome driven, where people look at mechanisms of resistance, finding new drug targets, or looking at the evolution and diversity of parasite populations. But of course, all genome driven research is limited by the functional understanding that we have of the genome. And that is a challenge because we are dealing with a highly divergent eukaryote. A third of its genes have unknown functions, difficult to transfect, not all stages culturable. So it takes a bit of an effort to systematically assign functions to these genes. And how to do that effectively has been what the lab's been about for the last few years. So there are a number of papers that over the years have generated resources for the community, molecular resources and data, and that's just illustrated here. So we've done various screens around the life cycle to ask different questions, created functional and transcriptomic data and transcriptomic atlas that we contributed to, etc. So lots of data that need to be hosted, displayed, and integrated. And what we can now do as a result of that, we can cluster genes not just by their expression patterns, but as shown here, for instance, by their functional data that we've collected from all these screens. So we are gaining insights into drug and vaccine targets, which we can predict systematically from this, and our colleagues, we can discover unique biology, and we can do things like build genome scale metabolic models and make them stage specific through our genetic data. And I've got a computational biologist in the lab who's got a bit of experience with that, for instance. So what we hope to do with the data platform now is find a way of hosting the various data types that we are creating. And we have a database going here that we are containerizing. Vikas is working with Hamza Imran Said at the moment, and we're hoping to get this database hosted soon through SciLifeLab. And there we have on the left a part of the database that shows and allows and shares us to allow reagents. Then in the middle, phenotypes, and integrate them with transcriptome data. And we're also talking with Michael Anton from NBIS to see how we can host and visualize our stage-specific metabolic models. So that is all work in progress. This is the team working on this. I'll leave it at that. Thank you very much.
Thank you very much. Your connectivity held out, so that was great.
It's running through my phone. This, wonders of technology. Here we go.
There's always a way. Where there's a will, there's a way, right? Okay, so we have a few minutes for questions for our speakers. So I know a few different ones came in chat directed to different ones of us. So I see one from Marcella, for example, about connection between the data platform and the SciLifeLab, and if we are talking to them. Yes, we are talking to them. So hopefully, obviously, they're currently building their resources over at the SciLifeLab Training Hub. And we hope to get events and training information in from them. So I'm actually going to be discussing that in a bit more detail with them next week. So looking forward to that. And what were the other ones? So by the way, you can, if you'd rather unmute yourselves for questions or raise your hands, and that is entirely within your power. So please feel free to do so. And then just another one in the chat from Samuel Lemper. So about how the platform will support clinical genomics use cases. I don't think we have just one way of doing this, perhaps, and it would be good to discuss more directly with those users specifically in order to be able to work out how best was to do that. So that's how we've typically done this with our use cases so far. And just scrolling down. So I think a couple of questions for you, Aileen, on the RDM guidelines about different resources that can be linked. So Joel mentioned the information from the data sharing policy. Joel, I don't know if you'd rather say out loud or that's sufficient.
Sure. So you're mentioning this research data management guidelines as a community effort, and we sure did that in IDA to support the collaboration arena for medical imaging diagnostics AI. So a blocker was access to good training data. You need tons of it for good AI, or otherwise you can't get anywhere. So that was obviously the blocker for progress in IDA. So when I came aboard that in 2018, it took me a year to compile the first version of this policy that I've linked there, and it's been developed ever since with examples from real life. How did people do anonymization of actual data sets that are being shared through the IDA Data Hub of clinical imaging and associated metadata? And how we are sharing that with the world, and how you should write your ethical review application in order to get it granted, to have permission for sharing, and so on and so forth. So we did a legal analysis and drilled down to original statements in official law texts. So this is an actively developed community resource that has been a tremendous enabler for IDA Data Hub, and we will happily see it shared with more communities. It's the same law, it's the same ethics, it should be the same also for other disciplines. So I suspect this applies to other communities in Sweden and in similar countries. If you have a freedom of the press law and official secrecy act, and if you work under GDPR and so on and so forth, this should apply. So I think it should be relevant for the SciLifeLab RDM guidelines.
Yeah, thanks. Definitely sounds like a good thing to add there. So we'll add it on the to-do list. And the next one, comment from Rosa, was also for me, I guess, linking to resources and the local universities and so on. We do have one topic where we try to link to university RDM resources, but I mean, you know best what you have, right? So again, feel free to suggest things and we will happily add them.
I think the last question we have in the chat is from Samuel Lamper. I apologize if I'm pronouncing your name incorrectly. I don't know if you'd rather voice it or have me voice it with your suggestion.
Yeah, well, I can maybe just add a little bit. I was just thinking that maybe as a complement to the guidelines, it can be good with some concrete best practices or like tips on good strategies for structuring your work so that it's reproducible and traceable from the start, because I feel like that's often the challenge. Maybe you do your project and then in the end, when you're supposed to publish the data, then you're starting to look at all those guidelines. And then maybe it's too late to even figure out what parameters you used for some software, etc. So that was just a thought that could be a complement, perhaps.
Yeah, I agree. That's an excellent point. Start to think about these things early and it will make it easier. And we do have some training material that covers that aspect that you can also find through the guidelines. But I also think it will be nice to look at the paper that you shared there and see if
I think it has been. Yeah, I think it has been shared in one of the courses actually.
Yeah.
But maybe just summarize the kind of top links.
Great. I think that was all the questions we had in the chat, but we have a few minutes left if anyone wants to get their questions in now. So perhaps no further questions right now, but if anyone feels that they want to reach out about the platform, about any of the services, we'll be making the recording and everything available. Obviously, there's the support emails for each of the services on the service pages, so you can contact them more directly, and reaching out to us on the data platform is always an option through either our data center social media or our general contact form on the webpage. So with that, I will let you all go. Thank you very much for joining, and I want to thank all of our speakers today. Really appreciate you joining and speaking here. I will inform everyone when the recording is available. Okay, so thank you guys.
Thank you.